My name is Georgia Atkin-Smith and I'm a senior postdoctoral researcher working at WEHI, formerly known as the Walter and Eliza Hall Institute in Parkville in Melbourne.
And I'm Associate Professor Edwin Hawkins. I'm a laboratory head at WEHI as well.
Tissue damage during disease is caused by programmed cell death through either apoptosis or necroptosis or other kinds of cell death programs. And typically it's been really hard to detect in vivo, and mainly due to the lack of access to those areas where damage is occurring during disease. Cells of all different statuses are able to generate subcellular cargo we know as extracellular vesicles, or also known as EVs. And EVs have many different functional roles in vivo through aiding processes like intercellular communication and also the efficiency of cell clearance.
Many years ago, we did some work at Imperial College in London, driven mainly by a PhD student at the time, Delphine Duart. And what he noticed is that in acute myeloid leukemia, a type of blood cancer, we saw dramatic remodeling of blood vessels in the body. What that led us to believe is that there was some sort of process to process and deal with that cargo that was released from those vessels. What we didn't know was the context of how it happened in disease and how the body responded to that pressure. So the hypothesis was that this remodeling of the blood vessels and processing of this tissue damage was an active process. And the idea was, once we could understand how it worked, we could not only understand the fundamental biology behind it, but also understand if these sorts of EVs or this waste that was caused during this process could be detected in the blood. The problem was we didn't understand how fast it happened, any of the dynamics, where it happened, or what was the biology that would drive it. So the methodology that Georgia and I applied was to perform what we call intravital imaging. This is a process where we use 2-photon microscopy to look at dynamic processes occurring in place. And in this case, the bone marrow, which is a notoriously difficult area to image. So what we did was we developed a method where we could perform surgery, look at bone marrow over multiple days during development of blood cancers and normal homeostasis. So that meant for the first time, we could watch this process of this cargo getting generated, understand all the dynamics behind the process, and make fundamental measurements about timing that would stand the test of time, not only informing our ideas and hypothesis, but setting a benchmark and a groundwork fundamental level for the whole field to work off for history.
By using this imaging technique, we were able to visualize for the first time the generation of these extracellular vesicles from blood vessels in the bone marrow. And we found that this was highly elevated during blood cancers, such as acute myeloid leukemia. And so these EVs were being generated and released into the blood. And so we found that by simply taking a sample of blood from our mice models, we were able to detect this significant increase. And so through collaborations with researchers and clinicians at the Peter MacCallum Cancer Centre, we've now begun to assess the blood of AML patients to see whether we can reproduce these elevated levels of EVs, which indicates the extent of tissue damage observed during cancers.
And I think what a great thing that Georgia showed in her study was that this might happen in a local area in the bone marrow, but these EVs could be released into the blood and detected a long way away. So what that means is we think that if AML start to relapse, which is when they evade therapy, and cause a new foci in the bone marrow, that in a very distal site like the blood, we can then take an accurate measurement and get an idea about what's happening before it becomes a problem for the patient.
This is an extremely collaborative study. In particular, we've had a huge amount of help from researchers at the Trobe Institute for Molecular Science. There they have established zebrafish models where they can perform imaging to see zebrafish biology. And so we've been able to, again, not only show this formation of EVs in a mouse model, but also collaborate with La Trobe to visualize this process happening in fish as well. But we've also had incredible collaboration from researchers at the ONJ, the Florey, and as mentioned also at Peter MacCallum's Cancer Centre.
I think we can look at that at two parts. So the first one is a practical test. So what we want to do is understand how many of these processes occur in different types of blood cancers and other cancers in general that interact with the bone marrow, and understand when these distal packages like extracellular vesicles can be used as a readout for, I guess, general vasculature health. This might even extend into autoimmune diseases with vasculature problems, so we think that that's really important. The other part is the measurements that Georgia made in her study are the first of their kind, and they will set a benchmark for science all around in this field. And what that means is in any case now where we see a perturbation in this, and we can measure it, or other groups can measure it, we've set the steady state level that they'll always be able to compare it to. And I think that's really important in the context of EVs. They are not extremely well understood. It's still a young field. And that means that really what we've said is the level to work from here to understand when anything's going wrong in vasculature health.
And to add on that, as Edwin mentioned, the field of EVs has been largely built on in vitro studies, as they are obviously small in size. Performing those in vivo models where you can visualize them has been really challenging. And so that's why we're so excited about this study, as it provides one of the first examples of where these EVs are being generated through this active biogenesis process in vivo. And we really hope that this can provide this foundation to extend this rapidly growing field of extracellular vesicle research.